Hi, my name is Gustavo Correa, and I am a PhD student at the University of California, Riverside. Today, I will be talking about the development and testing of a novel automated insect capture module for sample collection and transfer. This work is in collaboration between the UCR Autonomous Robots and Control Systems Lab, as well as the UCR Ray Lab. As robotics technology continues to mature, we are now interested in seeing how we can use autonomous mobile robots for in situ sample collection, handling, transfer, and testing. Amongst different diseases, the spread of arthropod-borne diseases is a serious concern faced by public health officials and the medical community at large. One question we ask ourselves is, how can we use robots to detect and track the spread of disease? Specifically, in this work, we are targeting vector-borne diseases, which are infectious diseases or illnesses transmitted through insects such as houseflies. There are several bottlenecks in the tracking, modeling, and prediction of disease spread. The first being the frequency at which we can sample insect vectors in environments that are prone to propagating disease. The second is the amount of manual labor needed to set up and retrieve surveillance device like insect traps. And lastly, the return time in analyzing insect samples and determining if an infectious disease is spreading in a region. So our goal in this work is to leverage autonomous robot technology to tackle these bottlenecks and speed up the process of collecting samples from the environment. As we build upon this work, we have the long-term vision of enabling rapid and large-scale sample collection. In critical situations, it is also important to reduce potential risks to human personnel that are tasked to collect and handle samples. And ultimately, it would be very beneficial if we had the capability to periodically sample insects in changing environments to track ecological changes. In our review of existing technologies, several insect traps exist that utilize ultraviolet light or other insect attractants, such as odorous food and baits to attract and capture insects. Some drawbacks of these devices are that they tend to be heavy and are designed to be permanently placed, such as the mosquito traps used in Microsoft's Project Premonition. We are motivated to design a trap that serves as a means for capturing, retaining, and keeping insects alive during aerial transport, and preferably a trap that can be transported with aerial robots available for purchase by the average consumer. Our contribution is the design, fabrication, and testing of a novel automated insect capture module, or ICM for short. The ICM is small, lightweight, and low cost. These characteristics enable the trap to get airborne via an aerial robot. Our main objective in the design of this trap is to engineer a structure that can capture, retain, and keep insects alive during airborne transportation. Our design features modular trap panels and insect entry channels. The ICM consists of the actual trap shown in red and the electronic space shown below. The entry channels can be interchanged to accommodate different insect types. Inside of the ICM is a UV LED core, which can also be easily removed from the bottom of the trap. And lastly, servo motors can be controlled to open the panels for easy access during maintenance. One critical design parameter of the ICM is the type of insect attractant used to capture insects in the environment. We selected ultraviolet light as opposed to odorous food and baits, since it is highly controllable and easily configurable. Although behavioral studies and literature suggest that UV light is the most attractive to houseflies, we could also utilize the same technology to attract other insects. The ICM features a UV LED core, which can be reconfigured with LEDs of different wavelengths and different LED arrangements. The electronics bay also contains a BigoBone blue used to remotely control or flicker the LED core. We place temperature and humidity sensors inside and outside of the trap to monitor environmental conditions during insect capture and transport. A small night vision camera is placed inside of the trap to monitor insect activity near the entry channels. With these sensors, we can assess the trap's habitability in different environments. We conducted 4 experiments to assess individual components of the ICM and its performance in near real-world testing conditions. For our first experiment, our goal was to determine the optimal entry channel diameter. so that the trap lets flies in, but does not let them out. We targeted these tests for flies with an average body width of 5 millimeters. For the first step in our experimental procedure, we designed entry channels with varying inner diameter from 3 millimeters to 7 millimeters. We performed entrapment and retainment tests with groups of 5 flies, where the goal of the entrapment test was to see how likely the flies were to enter the trap, and the goal of the retainment test was to see how well the trap could keep the flies inside. To evaluate the performance of the entry channels, we utilize the capture rate, which is the product of the entrapment and retaining rates. We determined that 4 millimeters is the optimal inner entry channel diameter with a capture rate of 87%. For our second experiment, we wanted to determine a suitable UV LED core configuration for housefly capture in lighted and dark environments. Literature suggests to use UV LEDs with wavelengths between 300 to 400 nanometers, since this is the most attractive to male and female houseflies. In our experimental procedure, we constructed various LED core configurations. We then placed the ICM and 50 flies into the insect cage. We turned on the UV LED core for 2 hours, where after the capture period, we counted the number of insects captured inside of the ICM. We then compared the number of insects captured in lighted and dark environments using each of the different LED core configurations listed in the figure on the right. And our preliminary results show that the ICM is capable of capturing insects in controlled environments, and this may have been because the UV light is more concentrated in an environment without external influences. Additional data is needed to confirm the ICM's performance in different environments. For our third experiment, we wanted to assess the performance of the ICM mounted atop of the quadcopter during takeoff, flight, and landing. We also wanted to assess the survival, mortality, and escaping of houseflies contained within the ICM during flight. For our experimental procedure, 50 flies were inserted into the trap, which was fitted with 4 millimeter channels. After mounting the trap on top of the quadcopter, we commanded the quadcopter to fly at preset paths at 5 meters per second and 2 meters per second for 5 minutes. Once we dismounted the ICM, we counted the number of houseflies that remained alive inside of the ICM. To evaluate the performance of the trap during flight, we wanted to observe any structural damage and loss of flies. And for our results, 95% of flies remained alive in the trap after transport, which was a great indication that flies can survive inside of the ICM under turbulent airflow. Moving forward, the ICM also has potential for transporting more flies. For our last experiment, we combined aspects from prior experiments to emulate insect capture and transport in a near real-world environment. These experiments were conducted in a greenhouse which was lined with tarp to block exterior light. We placed the ICM together with the robot inside the center of the room. We also placed lures inside of the room to emulate external influences. After releasing 100 flies into the room, we turned the UV LED core on for 2 hours, after which we commanded the quadcopter to hover for 30 seconds to emulate insect transport in turbulent conditions. At the end of the experiment, we counted the number of flies captured inside of the trap, and we also observed insect activity inside and outside of the trap during capture using the sensors mentioned before. Around 5 to 7 flies were captured after 2 trials. To conclude, the ICM captures, retains, and keeps insects alive during aerial transport. This device can be manufactured with typical benchtop tools such as 3D printers and laser cutters. Preliminary results for capturing and retaining mosquitoes provide an initial indication for developing a general-purpose insect trap. For our future work, we would like to perform an evaluation of species-specific lures, determine insect survivability under fast flight, and quantify energy efficiency in remote operations. Thank you for your time, and please reach out for any questions.